as soon as this anther sheds the pollen grain can go and fall on the stigma. Now, if that happens, what will happen? Self-pollination will occur. Where is the polar nuclei present? At the central cell. How many polar nuclei are there in the central cell? 2. So, 2 times fertilization will occur. By artificial hybridization, the desired pollen grains are used for pollination and the stigma is therefore protected from contamination. Hello everyone, a warm welcome to this session on concept class for class 12th biology. I am Dr. Divya, biology faculty. So in this session, we'll learn about the outbreeding devices, that is how to prevent self-pollination and achieve cross-pollination, and also we'll learn about the pollen pistil interaction, and also we'll learn about double fertilization event. So first we'll learn about outbreeding devices. So outbreeding devices are usually achieved in bisexual flowers. That is, naturally the flower itself will develop themselves in such a way that self-pollination is prevented to a maximum extent and cross-pollination is promoted. Why self-pollination is prevented is because sometimes new traits will not develop in the flower. The same age-old trait that is there will be there in the flower. To get new traits from a healthier plant or from a healthier flower, they prefer cross-pollination. So therefore, outbreeding devices are developed by the bisexual flowers in order to avoid self-pollination and to promote cross-pollination, because sometimes because of continuous self-pollination, what happens? The quality of the seeds will become very poor. Say for example, in that same flower, that plant is suffering from some disease. Now the seeds that will develop there will also get that disease. So to prevent that, what happens is, if cross-pollination is preferred, at that time what happens is, from a healthy, stronger plant having dominant characters, when those genes come to this particular plant and then the seeds develop out of it, then it will get a new variety or a good quality seed will get set up. So in order to prevent poor yield and to prevent poor quality of seeds, or what we can say inbreeding depression, to prevent the occurrence of inbreeding depression, that is, sometimes the breeding capability of the plant can also get lost, or the seeds may not germinate at all because of its poor quality. So to prevent all that, flowers usually they develop this technique of outbreeding devices. Now what are the different outbreeding devices that has been developed by the flower? We can see here. This is natural, that is, the flowers itself have found a way to promote cross-pollination. Okay. So here the pollen release and the stigma receptivity are not synchronized. Say for example, I have plant 1. So here self-pollination means autogamy. In the previous session we learned, right, wherein the stigma and the anther that are present in the same flower, it is a bisexual flower, wherein the anthers, once it releases the pollen grain will fall on the stigma, right? So say for example, here I have a plant P1. Now here the flower is there in this P1. Now in this P1, what is there? Stigma is also there. So I'll write it as S, and anthers are also there, right? That is why it is a bisexual flower, because both the male and the female reproductive parts are present. Now, as soon as this anther sheds the pollen grain can go and fall on the stigma. Now, if that happens, what will happen? Self-pollination will occur. But the plant's idea is, I should not promote self-pollination. I need only cross-pollination to occur here. So what the plant does is, it will release the pollen and the stigma at different types. That is, it will release the pollen when the stigma is not mature enough. So the stigma may not be mature. It will be in a young age itself, but the anther will get matured very fast and will release the pollen grain. So that the pollen grain, even though it falls on that stigma, self-pollination will not get achieved. So that is one of the technique followed by the plant. And next is, they follow a process called as dichogamy. Dichogamy means anther and stigma are placed at different positions. Say for example, the stigma may be very lengthy and the anthers may be very small. For the pollen grain to go from here and fall on the stigma, it is very, very difficult, not achievable. So that is called as dichogamy. That is, the anther and the stigma will be placed in different position. That is one of the outbreeding technique. Next is self-incompatibility. What the self-incompatibility means? Sometimes what happens, two flowers will be there, but the pollen grains will not be compatible with the stigma. Though it may go and fall on the stigma, the stigma might produce certain substances which will prevent the pollen grain from germinating on that stigma itself. So like that and all, they'll show incompatibility. Or they may show unisexuality. Unisexuality means in one plant, the flower will produce only the anthers. In one plant, the flower will produce only the stigma, like papaya. Papaya is one best example. So that their self-pollination is not at all achievable. Cross-pollination is only possible. So that is what unisexuality, or it may become chasmogamous flowers. So what are chasmogamous flowers? In chasmogamous flowers, the anthers and the stigmas are well exposed, so that the stigma is so well exposed that it is easy for the stigma to receive pollen grains from the other flowers. So some flowers will exhibit chasmogamy. So these are the outbreeding devices that the flower itself follows in order to promote cross-pollination and to prevent self-pollination. Now there is one more thing that we human beings can do to promote cross-pollination. That is called as artificial hybridization. That how it is done we will see next. So before that we need to talk about pollen pistil interaction. Now, when pollen grains from the anther, they have to come and fall on the stigma, some interaction will occur between them. What is it is what we will study here. So, it is mediated by chemical components of the pollen interacting with those of the pistil. So, there are certain events that occur in the pollen pistil interaction. Following compatible pollination, pollen grain germinates on the stigma to produce a pollen tube through a germ pore. Now here, as I told you in the previous sessions, once the pollen grain falls on the stigma, they need to produce a pollen tube. So that is the pollen grain, it will produce a pollen tube, which is also called as the germ tube. Now this pollen tube will get produced only if the stigma allows it to. I told you, that is what I told you in outbreeding devices, incompatibility. Now, if the stigma produces a chemical which will not promote the pollen grain to germinate, then how will fertilization be achieved, right? So, here what happens in pollen pistil interaction is only the chemicals which are capable of promoting the germination of the pollen grain, if it is present on the stigma, say for example, only if enough amount of sucrose is produced by the stigma, then only the pollen grains will germinate and the pollen tube will get formed. So that is the first interaction. And the second interaction is the pollen tube grows and it reaches the ovary. It enters the ovule through the micropyle and then enters one of the synergids. Now what happens here is, if it is compatible, if the pollen grains and the stigma are compatible with each other, if they are in good terms with each other, then the pollen grain will germinate, it will produce its pollen tube. The pollen tube will enter through the micropyle, that is the opening. So this is the micropylar end, micropyle, and this is the chalaza. Now it will enter into and it will deliver the pollen grains towards the micropylar end. That is the compatibility. That is the pollen pistil interaction. Now what did we get to learn from so far? We got to know that the embryo sac is ready to receive the pollen grains. Then the anther is ready. It has released the pollen grains. Now the pollen grain has traveled through the process of pollination and it fell on the stigma. Now the stigma, if it is compatible with the pollen grain, it will allow the pollen grain to produce the germ tube. Now compatibility is there. So therefore, the pollen grain has produced the germ tube. The germ tube has entered, it has increased its length and entered into the micropyle. Once it has entered into the micropyle, it would deliver the male gametes that is present in it to the synergids. Next, what happens? We'll study in post-fertilization events in the next session. So this is about the pollen pistil interaction. Interaction is nothing but the compatibility between the pollen and the pistil. So we'll talk about artificial hybridization now. I told you artificial hybridization is a man-made outbreeding device. Like there what we talked about in the previous slides were the outbreeding devices that was developed by the plant itself to prevent self-pollination and promote cross-pollination. Now artificial hybridization is man-made, wherein we ourselves, we promote cross-pollination by preventing self-pollination. How we do that, we will look into. So by artificial hybridization, the desired pollen grains are used for pollination and the stigma is therefore protected from contamination, that is protected from unwanted pollen grains. So this is done by two important processes. One is emasculation and bagging. Emasculation. In emasculation means masculine, something related to the male. So what is removed there? E means removal of something. So here anthers are removed. The male reproductive part of the flower, anther is removed from the flower bud. Say for example, here I have a plant. There is the flower. In this flower only, the stigma is also there and the anther is also there. So this is the stigma, this is the anther. Now the pollen grains, when it is produced like this, the pollen grains can fall on the stigma, and if the stigma is compatible, the pollen grains can germinate on the stigma. It can produce the germ tube. The germ tube will enter through the style of the stigma and enter into the ovary, and in the ovary it will enter into the micropyle of the ovule, and then it will deliver the male gametes there if it is compatible. Now we don't want self-pollination there. We have to prevent it. So what scientist does is, he will completely remove these anthers. When the anthers are removed, then self-pollination is not possible. But pollination will occur because pollination from some other flower, the pollen grains from the flower which we want, which is having good characters, we can take that and we can dust it on the stigma to achieve pollination. So that is what is done here. So once the anthers are removed, what should we do is, we have to, that is first emasculation, that is the anthers are removed from the flower bud before the dehiscence using a pair of forceps. After emasculation, what we do is, we take the pollen grains from the desired flower, we dust it onto the stigma and then we cover the stigma with a bag. That method is called as bagging. Why bagging is done? Because otherwise undesired pollen grains from some other flower might come and fall on it. No, to prevent that, bagging is done. So that is called as bagging. So one is emasculation and the second one is bagging. That is the emasculated flowers that are there, they will be covered by bag to prevent contamination of the stigma by unwanted process. So two methods are followed in artificial hybridization. One is this one, that is emasculation, wherein the anthers, if you can see these anthers, these anthers that are there, they will be removed using a forceps. Then what will be done? Onto the stigma of this flower, from somewhere, whatever pollen grains we need, of our choice, it should be of the same species of our choice, we will bring it and dust it onto it. Once it is dusted, we will bag it, cover it with a bag. Bagging is done and it is left. Now the process of fertilization will start here, and how the fertilization will occur and all that we will study in the next session. That is the post-fertilization event we will study. So next, moving to double fertilization. This is what we need to know here. The fertilization process that occurs in the flowering plant is called as double fertilization. Why it is called double fertilization, we will look into. This is one of the important questions. Artificial hybridization is one of the important questions. Double fertilization is very important. Pollination, all that is very important in this chapter. So double fertilization occurs after the pollen tube. So now what has happened? The pollen grain has come and fallen on the stigma, and if the stigma allows, that is, if there is compatibility between the pollen grain or the stigma, or to make it easy for you, I can say, if the stigma likes the pollen grain, then it will allow the pollen grain to germinate. The pollen grain will produce the pollen tube, or it is also called as the germ tube. The pollen tube will increase in its size and it will reach the micropylar end. Once it enters into the micropylar end, it will deliver the male gametes. So, can you see here the male gametes are being delivered. So, this is one of the male gamete. This is the second male gamete. Two male gametes will be delivered. Now, the male gametes will move where? To the synergids. So, this is the synergids. So, this portion itself, this portion that is there now, we have drawn, it is expanded here. That's it. Okay. So, the male gamete. So, this is one male gamete and this is the second one. Two are delivered here. So, the next, after the delivery of the male gamete, what will happen? One of the male gamete that is there, it will fuse with the egg cell. So, egg cell you remember, no? Can you see here one more? Because we learned that towards the micropylar end in the embryo sac, 3 cells will be present. They are called as the egg apparatus. In that 2 will be synergids, 1 will be the egg cell. So, one male gamete that is there, it will go and fuse with the egg cell. So, one fertilization occurred, right? Why? Because that is, so okay, to make it clear for you, pollen grains in that 2 male gamete will be present. Okay. So, this male gamete will be delivered into the embryo sac. Now, in this embryo sac, what is there? Here, 2 synergids are there and this is one egg cell. So, one of the male gamete that is there, it will fuse with the egg cell. First, one fertilization will occur. So, what is fertilization? Fusion of the male and the female gamete. So, one male gamete will fuse with the female gamete. One will fuse with the egg cell. One more male gamete is there, no? That is being released by the pollen grain into the synergid. That will go and fuse with the polar nuclei. Where is the polar nuclei present? At the central cell. How many polar nuclei are there in the central cell? 2. So, 2 times fertilization will occur, wherein one male gamete will fuse with the egg cell, other male gamete will fuse with the central cell, nucleus present in the central cell. Because 2 times fertilization is occurring here, this process is called as double fertilization. So, now you might have understood why it is called double fertilization, or it will go and fuse with the nucleus, which is called as the primary endosperm cell. So, it will go and fuse with the, or we can call this as the primary endosperm cell. So, that is called as double fertilization. So, this is about double fertilization. So, this was about the session. So, in the coming session, after the process, that is, once the fertilization is occurring, what will happen, we will look into. That is, after the fertilization. So, fertilization is achieved here. Till now, whatever sessions you learned in this session, you got to know that fertilization is achieved. What type of fertilization? Double fertilization. Wherein one male gamete that was released by the pollen grain, it went and fused with the egg cell. One more male gamete which was released, it went and fused with the central cell. That is the polar nuclei present in the central cell. 2 times fertilization occurred. Now, after that, some structures will get formed after the formation of the fertilization. Like the seeds have to develop, the fruit has to get formed and all that. That how it happens we will look into. That is, we'll learn about the post-fertilization structure and events. Post-fertilization means after fertilization what all will happen. In that again we'll learn about the endosperm and the embryo in the seed, and we'll learn about the structure of the dicotyledonous embryo and also about the monocotyledonous embryo. So, we'll learn about this in the coming session. So, I hope you found this session useful and you were able to understand. So, we shall meet again in the coming session discussing the following topics under the same chapter. So, see you in the next session. Thank you.